Thanks, Mark, and I would thank Monsoon and Rudy, but he cut the time of my talk down, so there isn't time to do that. But thanks a lot, and thanks for everybody turning up today. A lot of very familiar faces in the audience, so good. So I'm going to give some of you an update on where we are in our thinking with race, and hopefully introduce it to some of you as a new story. That's why you get the pointer working, which I can't. So let's have a look. Right, so just a very, very quick corporate snapshot. So market cap just under 250 million. We've got just under 15 million in the bank as of September. We've got an R&D tax credit coming up, which I can't tell you how much that is, but that's going to swell that number quite significantly. And one of the things we did last year was make sure that we can get to the end, get to some meaningful clinical data with the money we have, and we've made sure that we're able to do that. So everything I'm going to talk about today is fully funded out of current reserves. We also have the piggyback option. There was a bonus option scheme, and there were 3 piggyback options that people converted, and very grateful for the support we had that most of them did convert. So we have potentially $25 million of additional piggyback options that will convert. They're in the money, not by as much as I would like, but they're in the money, and they run out till May the 23rd, 2026. So I'm going to go a bit faster than I normally do with this. Management team, we've been building up the team over the past really 18 months, which slightly predates my joining. Daniel Tillett, who was CSO, cornerstoned an investment round, is CEO. There isn't a person on the planet who knows more about the drug I'm going to tell you about. And he's doing a great job as CEO, and we're thoroughly enjoying working together. Michelle Rashford, very quickly, a lot of clinical experience with Big Pharma. So Sophia Moscovis, also big pharma experience. She's a VP of operations. Mike Kelso is a gun medicinal chemist, and he runs a very, very productive team at the University of Wollongong. And he's a full-time employee, but he manages a team. We rent space down there. And Marinella Messina is the last person I want to mention. She's got a lot of experience in CROs, so actually running clinical studies. On the board, Daniel, myself, and we were recently joined by Serge Scrofani, who's in the audience. Serge's got a very a fantastic background in business development, having worked at CSL in a number of very, very senior roles and being involved in a number of very substantial multi-billion-dollar transactions. So it's great to have Serge and I worked at AMRAD 20 years ago, which is quite embarrassing, but anyway. So great guy, go and grab him if you can afterwards. So I'm going to introduce this by talking about cancer survivorship, and I was up at the Clinical Oncology Society of Australia meeting last week. I was actually very, very surprised. The majority of the talks were actually on the subject of cancer survivorship. So we've got a lot more people with cancer, growing population, but also the oncologists are getting better at treating them, and so we're getting a lot more patients who are functionally cured or in long-term remission. And that's great, but the problem that's emerging because of that is a lot of these people have a lot of deficits because of the chemotherapy. And when I joined RACE, the focus was entirely on the heart. We've expanded that to the cardiovascular system, and we're now starting to look at other parts of the body, because we think mechanistically the protective effect that our drug has on the heart may extend elsewhere. But the problem, which I'm going to outline, is that these chemotherapeutics are very, very toxic and do permanent damage, particularly to the cardiovascular system. We do use our cardiovascular system to 100% of capacity. Running up a hill, running for a bus, down the gym, you really notice if you lose 15-20% of that capacity. Kidneys may also be affected, but you can lose a lot of kidney function before it becomes a real problem for you. So I'm talking about the anthracyclines. I won't go through the slide, but they're very widely used, and in fact the use is growing. So these drugs have been around since the 60s, but they're still widely used across a range of different indications and a lot of different combinations. So whilst they're like wallpaper to a lot of people, these drugs are still the backbone of a lot of regimens in oncology. And these drugs, particularly the anthracyclines, do cause a lot of cardiotoxicity. And in fact, the solution to dealing with that is to just reduce the amount of drug you give to the patient. So the amount of doxorubicin, for example, which is the most widely used anthracycline, is capped at 4 cycles of therapy, and then you stop. The clinicians stop. If they go further than that, then they're increasing the risk of cardiotoxicity and causing heart failure, maybe not today, but quite possibly down the road. So the issue we have is, well, first of all, there are a lot of patients who can't be treated because they have cardiovascular risk factors, which would be simple things like high blood pressure, which is very common in an aging population. So a large number of people who can't be treated because of the risk of cardiovascular side effects, and then you've got a large number of people who are being underdosed because they're stopping giving the drug, the doxorubicin, not because it's not working, but because of the fear of cardiotoxicity. So the opportunity that we see is, well, first of all, it would be nice if these patients weren't harmed as much as they are. And a lot of them don't actually know, certainly not ahead of time, that this is going to happen to them. But then we can maybe able to treat patients who would otherwise not be able to receive these potent anti-cancer drugs. And obviously the real goal at RAISE is to enable the oncologist, if a patient is still responding to a drug, to continue to give that drug without fear of those permanent side effects. So that's what we're all about. And so let me switch now and actually talk about what we're doing at RAISE, which is developing bisantrene. So as those of you who've been following the story will know this, but the drug was being developed in the 80s by a company called Lederle that was acquired by Wyeth and ended up at Pfizer. And they developed this drug bisantrene. It went through well over 1,500 patients in clinical studies, including 9 phase 2 and phase 3 clinical studies, and these were in solid cancers like breast cancer, as well as hematological cancers like leukemia, and very, very active. But the problem was that the drug was very insoluble. So as soon as it got to physiological pH, it would crystallize, and that meant it had to be delivered straight into a major vein, delivering blood back to the heart. So there was a commercial reason that it didn't move forward. It proved itself to be safe for a cancer drug, and very efficacious with around 40% response rate in AML, something that we've repeated with some studies that RAISE has supported. It is active. There's a study that RAISE conducted across 143 different cancer cell lines across the major cancers. You probably can't read them all there, but they're all the big ones. So this drug has potency across nearly all of those major cancer types. And very importantly, when we put it together with doxorubicin, it increases the activity of doxorubicin because we're putting an anti-cancer drug with an anti-cancer drug, just another combination, and we get this improvement in efficacy. So in our upcoming study, we would expect to see hopefully some signs of enhanced efficacy, although small numbers of patients. But then very importantly, and this was a relatively recent discovery at RAISE, going back a few years, putting bisantrene together with doxorubicin actually protects the heart. And we understand the mechanism. I can't tell you what it is because we're going to publish that sometime next year in a peer-reviewed journal, hopefully a very high impact one. But what you can see here, and I'll point, this is baseline, this is looking at cardiac function. This is the controls at day 42. With bisantrene, there's a little bit of cardiotoxicity, but with doxorubicin there's significantly more. And then as we add more bisantrene into the doxorubicin 1-to-1 ratio, 2-to-1 ratio, we see that the level of damage is reduced. So it's really quite striking that you're putting two drugs together, both have toxicities, and yet we end up with less toxicity in the combination. I can't think of another example of that. And down the bottom panels, the middle one with the grey streaks in it, that's fibrosis. That's collagen being laid down as part of a healing process in the heart muscle. I don't need to explain why that's not a good thing. The right-hand panel is looking at the combination down the bottom here, is the combination of bisantrene and doxorubicin, and you can see it's almost looking like the untreated or the control. So very, very interesting. Okay. Now let's get back to the issue that Lederle had that led them to discontinue or not to commercialise bisantrene, and that was the lack of solubility. So RAISE has reformulated bisantrene into a new formulation called RC220. Importantly, once it's administered to a patient, the drug behaves the same way in the body, so it has the same pharmacokinetics and pharmacodynamics. You'll have the same amount and going to the same places as the original formulation. So that means we can rely, in inverted commas, on the historic data, the data that says that this drug is effective and is safe. So that's very, very important. It also brings us some new intellectual property, and there are some other IP filings that will be taking place in the future as well to really shore this up. And so that gives us a nice long patent life. And we're doing a lot of work now to understand the mechanism, and in particular, very, very interested to see if we can actually arrest or reverse that aging effect that chemotherapy has elsewhere in the body. So starting to branch out, not just looking at the cardiovascular system. We've completed all of the preclinical tox. All of the documents have been done, ready to start the clinical study. We're just waiting for the first ethics submission to go in, and then we'll be hopefully getting the first patient on in the not too distant future. So in terms of the pipeline, we have supported some studies in AML, acute myeloid leukemia, which was a clinician-initiated study, again seeing a very good complete response rate in a very, very heavily pre-treated group. And that has sparked some interest in doing further AML studies, so again, investigator-sponsored. But the study that we're really interested in is the RC220 cardioprotection study. So there'll be dose escalation of bisantrene on top of doxorubicin, and then we'll be expanding and really looking in detail at all of the cardiovascular biomarkers, all the cardiovascular performance, so measuring VO2 peak and the like. So that's going to be, I think, a really important study. And if we show that cardioprotection in man and safety, obviously, of the combination, although there's really no reason to expect that we're going to be surprised there, but we have to do the study. That's going to, I think, squeeze a lot of any remaining risk out of the program. The market potential, well, it could potentially be very substantial. As I said, $1.5 billion sales of doxorubicin a year, and that's at $100 a vial. Our market research said that if we do manage to show cardioprotection and an increase in efficacy, that we'd be able to charge maybe $15,000 a vial. So you really only need a very, very small percentage of the current doxorubicin use to switch to combination use to drive some very, very big figures. So we're excited at that opportunity. Clearly we need to think about how we access that very, very big market, and so we're thinking, looking at things like regional deals, for example. So in terms of recent and upcoming milestones, I think we've achieved a lot in the past 12 months. I'm thinking past 12 months because the AGM's on Monday, so we're going to be talking about the past 12 months. But I think moving the program forward, stabilising the team, we did have quite a few changes at the board and management in the previous 12 months. That's really stabilised. We've had one person leave the company in that entire time. That's good. I think in the US it's typical to have a 30% staff turnover per year, so we're doing very well. So we've cleared all the hurdles we need to be able to get into the clinic, and then really now the key one is just getting that trial started. As I said, there's really no impediment. All the documentation's done, and we're just in the process of signing up the clinicians and moving forward. And then there'll be a lot of data coming out of the trial. It's an open label trial. It won't be weekly or necessarily monthly, but as we get data that we think is of significance, we'll be able to release that to the market. One of the advantages of an open label study. And then we are going to be filing an IND. We don't need an IND, but we're going to do it because that then means we have a gateway into the US if we want to collaborate with an oncology group there, for example. And we are thinking about pediatric uses of RC220 bisantrene. And it's really good to know that what we're doing is to that standard, so we don't have to read it. Right? Really, thank you. So that brings me to my final slide.
Oh, well done.
So I think the way that I look at this is a substantially de-risked asset. It's not without risk. We've got to establish safety in combination with doxorubicin, and then we've got to really show that it adds cardioprotection, because without that, I think we're going to struggle. But all of the data we have is highly suggestive that's going to happen. So it's about 3% chance of getting to market if you're setting off with an oncology drug at this stage, so just getting in first into man. I don't know exactly where the risk is now, but a lot of that has been squeezed out. It's certainly lower risk than 3%. The other thing that's really important is we are solving a real and increasingly recognised problem, and that is the cardiotoxicity and the long-term impact of chemotherapy. And this is particularly so in kids. There's been a lot of studies done in children for obvious reasons, but now we're starting to see more data coming out in adult populations who are affected. And there's this ageing thing. If you think about it, there are a lot of drugs that you can't give to older people because they can't tolerate it. And the way I'm starting to think of that is they don't have enough lifespan left to be able to take a hit at 10 to 15 years. So maybe that's what's going on. The other thing is that the market, in a sense, already exists. It's doxorubicin and the use of other anthracyclines. So our job then is to convince people to use it in that combination. And hopefully, if we get data around cardioprotection, aging more broadly, other tissues, I think people would put this drug in the "why wouldn't I use it" camp. And hopefully we can get a bigger market share than that we need to get that 7 billion a year sales. And the other thing, again, which is great, is we know how this drug is likely to behave in the clinic, and that helps a lot with planning. So I'll leave it there. Rudy waved at me a couple of minutes ago, so I'm probably for the high jump afterwards. But thank you very much.
I thought so. We saw that anthracycline was in the previous graph was really heavily used in Japan and China. Why is that? We saw the US was barely a blip on those graphs.
Yeah, well spotted. I'm not absolutely convinced by that data. We need to do more research, particularly in China. I mean, obviously you've got a very, very large population, and a lot of people aren't necessarily in centres where they're going to be able to get targeted therapies, immunotherapies and the like. So it could be that, particularly in regions, that some doxorubicin is still being very, very widely used. But I mean that data is very, very striking. So it's our independent data of us. So I'm a bit suspicious. Equally, we had some data showing relatively modest use in India, which doesn't seem to fit with that pattern. So I think one of the things we'd like to do in the coming years is actually get some people on the ground there and do a proper analysis of how's it used, who's using it, who are they giving it to, what are the trends, what's happening in terms of competition. But certainly on that basis, some sort of deal with a Chinese company at some stage would look like a very, very good thing to do. So we would definitely do that through partnership. We're not going to try and do anything in China ourselves.
Can you give us an update on where the trials are up to, when we can expect some updates on that, and what sites, etc.?
Can't name a site because we're still looking for the lead site. So that's in process. As I said, there's nothing on the race side that needs to be done. It's just about getting a site signed up. We've been through a period when there have been a lot of international conferences like ESMO and ASH is coming up, although that's less relevant to solid tumours. But it's taken a bit longer than we hoped. But there's no underlying problem, which I think is the important thing. But these things are always a selling job. You've got to get out there and you've got to convince people. And on first blush, this is chemotherapy. And, that's not that exciting. So we have to go out and sell. We have to go out there and convince people. And just last week, I was talking to a clinician, and he actually treats older population. And I said, well, I bet you don't use doxorubicin. I use loads of doxorubicin. I love it. So his name's in the hopper now as well. So it's just a matter of, the issue you have, it's very frustrating, is we're not in control of somebody else saying yes and signing up and putting the ethics in. We have to go around and talk to people, and then it starts moving. But hopefully not too far away.
Peter, I'll just ask you about your tax rebate.
Well, I've never had a question on tax rebates before, but yeah.
Well, you just announced that. Is it $20 million that you'll be potentially have as the rebate, or is that correct from the last 3 years?
No.
That's allowable. That's what it'll be 43% off the 20 million. Is that right?
No, not. I mean, this year, it's a reasonable number. We had quite heavy expenditure last year with the GMP manufacture, and then the preclinical studies. I mean, those preclinical studies were about $3 million just for those. So it's pretty heavy. Now the expenditure's settled down, and it's just going to be really clinical trial costs and a little bit of R&D. But so reflecting that quite high spend last year, we'll be getting roughly. I use 30% of the net loss is roughly speaking what you get back.
Thanks everyone for coming. Rudy and Monsoon, thank you very much for the invitation to be here. I'm excited to tell you the story about Island Pharmaceuticals. I think what you'll see from our story is while the indications are different from what we just heard about, the strategies are not that dissimilar. And I think that it's really a testament to sort of the idea when these companies got started is we didn't want to be an early stage R&D company. We want to focus on clinical trials. That's where the rubber meets the road, and that's really what we want to see, how our molecules work in clinical trials. Island Pharmaceuticals, we're obviously ASX listed. We are an antiviral therapeutics company, small molecule drug development company. We like to focus on viruses that are probably underserved. We don't want to be going head to head with the Mercks and the Gileads of the world. We think that there are many, many viruses. We all know there's many, many viruses where there's really no therapeutic agent. Vaccines are limited, and in the case of dengue, vaccines are actually really problematic. So as we, as a company, began evolving our strategy and thinking, what do we want to focus on? The assets that came to mind are those that come to us are small molecules. They come to us with clinical history. They typically would be have rapid paths to approval. We might have an open IND. We like the idea that they may be eligible for priority review vouchers. I don't know how often you talk about PRVs, but I know it's something you've talked about in the past, and we think that that's really important. A PRV, for those of you who don't know, is an incentive created by the FDA to really encourage people to develop drugs for underserved indications. We're pursuing what would be called tropical disease priority review vouchers, and in short, when you get this voucher upon approval of your drug, you get a voucher to get expedited review of a different program, and you can sell these. And the most recent ones have sold for about $150 million US, and we find that very, very attractive. Molecule called ESL 101. ESL 101, fascinating molecule. It has activity against a wide variety of flaviviruses. These are mosquito-borne viruses, so think dengue, West Nile, Zika, yellow fever. There are 400 million dengue infections a year. About 100 million of those require some form of intervention, and there is no intervention. You can treat the symptoms. There's no therapeutic for dengue or any of these other flaviviruses. The vaccines are challenging, to put it mildly. In some instances, they're actually dangerous. So they have significant problems with vaccines. We have a data package that's actually really fantastic. Work from Monash University that we're really excited to be partnering with Monash University on this program. We think that there is significant market potential for a molecule that could be potentially a preventative or a therapeutic for the dengue virus. I think it's actually really important to look at what we're seeing about the global cases of dengue over the last decade. Check out 2024. This is going down as the worst year on record for dengue infections around the globe, and we can talk about why, and I have a couple of slides we can address that. Global warming contributes to this. What we're seeing is mosquitoes moving into places they've not been seen before. They're bringing the viruses with them, and so now, if you look at the northern part of Australia, as you mentioned, or the southern part of the United States, we're seeing locally acquired dengue cases in these locations, which is a huge problem. We do have a pipeline expansion program in place. I'll tell you about a really exciting molecule we're very excited about. And moving forward with that. This is a slide I like to show you. This is an evolving slide. It changes nearly every time I give this presentation because what it shows you is dengue cases that have occurred, outbreaks that have occurred just in the last 3 months. And so if you look at Australia, there's a little bit in northern Australia. You can see an orange blip there. But Southeast Asia is always on fire. India is always on fire. Latin America is on fire. The United States, the southern part of the United States, Florida is having massive outbreaks right now. Florida is pretty warm, it's pretty humid, it's tropical. They also just had 2 hurricanes. So there's lots of water sitting around standing stagnant in Florida, and it's hot. Right breeding ground for mosquitoes and bringing the virus, the dengue virus. The fascinating one for me on this slide is look at California. I don't know who else been to California, but California is not tropical. Right, this is San Diego, this is Los Angeles. There's locally acquired dengue cases occurring in Los Angeles and San Diego, and that means these are not people who went to Mexico or the Caribbean and came home. That means the mosquito lives with you, and that's a real problem. And if we think it's a problem today, I'm going to tell you, as Mark previewed, It's a train coming down the track. This is where dengue is predicted to be endemic by the year 2050. Now look at Australia. The northern half of the country is predicted to be endemic. The southern third of the United States and the southern half of Europe is predicted to be endemic for the dengue virus. Again, vaccines are problematic. There's no effective therapy. We think that creates a huge opportunity for a company like Island Pharmaceuticals with a small molecule currently in phase 2 clinical trials looking at its activity against the dengue virus. So what's ESLA 101? Again, the analogy to what you've done at RACE. ESLA 101 was developed many, many years ago by a large pharmaceutical company. It was developed by J&J as a cancer therapeutic. It's been in thousands of people in clinical trials. It's been in 40 plus clinical trials. It's got a well-known safety profile, but it's never been approved for anything, not for safety concerns, but because it's not a very active cancer drug. So we obtained data. We were looking for a molecule that had anti-dengue activity and saw a piece of work from David Jan's lab at Monash University where they had screened libraries of thousands of compounds looking for agents with anti-dengue activity. Identified a molecule, characterized that molecule. I'll show you some of that data. And then they recognized this is fenretinide. It's ESLA 101, a well-known compound. So that was an aha moment for David Jans and for Bill Garner and I when we started this company, because we were very excited to find this work and recognize that there was a lot of work that had already gone into this molecule. And about 3 weeks after Monash published their work, Harvard published nearly an identical piece of work. They had screened thousands of compounds looking for an anti-dengue molecule, came up with fenretinide, what we call ESLA 101, did almost the same body of work. So when I put my scientist hat on, seeing academic labs independently validate work and arrive at the same molecule when they started with a very unbiased approach makes me very excited that what we're seeing is a real effect. So ESLA 101 has in cell culture activity against all 4 strains of dengue, West Nile, Zika, yellow fever. We've also looked at freshly isolated human cells, the cells that actually get infected with the virus, has excellent activity at protecting against infection there. There are animal models of both dengue and Zika infections where ESLA 101 is shown to be protective. And then recently we've completed a phase 1 clinical trial as a dose escalation study saying if we put ESLA 101 into humans in a dose escalation fashion, what's the blood concentration? What's the dose at which we're going to achieve a blood concentration that we think will be effective against this virus? I'll give you a little bit of a mechanism of how ESLA 101 is working, and I think it's actually really elegant. I'm also going to walk. So this is a host cell, a human host cell. When the virus enters the host cell, it really hijacks the protein-making machinery of the cell, and it converts all of the viral RNA into viral proteins, and they go on to make many more viruses. One of those proteins is called NS5. NS5 functions by getting in and out of the nucleus. It has to cycle in and out of the nucleus of the host cell in order for the virus to propagate. And it does so by binding to a nuclear import protein. So NS5 binds to a nuclear import protein. ESLA 101 prevents that interaction, okay? The very cool thing is if you look at the NS5 protein across all 4 strains of dengue and West Nile and Zika, it's nearly identical, which it highlights the fact that the mechanism of why we have such broad activity against all of these viruses. I think it's really, really elegant. Another piece of work that David Jans did here is looking at the concentration of ESLA 101 that effectively prevents viral replication of the dengue virus. So we're looking at increasing concentrations of ESLA 101 across the x-axis, and then the number of viral units, the live viral units across the y-axis. And the concentration he arrives at is 0.81 micromolar. And I'm just going to tell you that because that's a number that you can compare different drugs and their activity against a particular agent. But the important thing to know is that the concentration of ESLA 101 that we're dosing that we have dosed in our current phase 2 clinical trial we know is going to achieve a blood concentration about 5 times that dose. So we're well above what's needed to be effective against the virus, giving us a lot of confidence about our phase 2 clinical trial. This experiment is one that I became very excited about when I saw this. I know some of our current investors are very excited about this piece of data. This is an animal experiment. And it's a lethal animal experiment in mice. These mice are infected with a lethal strain of the dengue virus in the presence or absence of ESLA 101. So what we're looking at across the x-axis is time after infection, and on the y-axis percent survival. So at day 0, time 0, 100% survival. But if you look at day 5, These are animals that were infected but not treated, and 100% of the animals are dead. If you give them 1 dose a day of ISLA 101, you save about 20% of the animals. If you give them 2 doses a day of ISLA 101, you save about 70% of the animals, a very nice result. And in the drug development world, when I see dose responses like this, it gives you confidence that the activity you're seeing is related to the drug that you're giving. So we like this. The other thing I'll tell you is when you calculate the dose that we're giving or that we have given in our phase 2 study, compared to this, we're dosing it about 10 times. So it's already been demonstrated to be effective in this mouse study. So the phase 2 study, here's the thing I've been excited to tell you. So we're in the middle of a phase 2 clinical trial right now. The study that we're running is a challenge study. By challenge study, I mean we enroll healthy individuals, we infect them with an attenuated or a weakened strain of the dengue virus. We do this in a hospital setting, very well controlled setting, and then we see if we can either prevent the virus or if we can treat the virus if we administer ISLA 101 after infection. We're running this in New York at SUNY Upstate in Syracuse, New York. I wish like heck I could do it in Australia, but I can't. We're doing this in collaboration with the U.S. Army, and the U.S. Army dictates that the virus that they've developed only goes to SUNY Upstate. So this is a virus that was actually intended to be a vaccine, and it made people a little bit too sick. And so rather than discard it, the Army said, why don't we enroll some healthy individuals? Let's infect them with this, expose them to this virus, and see how people respond. Can we measure a dengue infection in a well-controlled setting? And they actually can. People signed up for this. People got paid to do this. You might not be surprised. But what it does for us is they now run between 30 and 50 individuals through their phase 1 studies. We have a cooperative research and development agreement in place with the U.S. Army, and they're giving us the virus. They're giving us all of the data out of the phase 1 studies that they've run, so it is a comparator database for us to compare our phase 2 results to. So the study that we're running, I've already teased it, we're running 2 cohorts. The first cohort, which we are well into, is a prophylactic cohort. So in this we enroll healthy individuals, we pre-treat them for 3 days with ISLA 101, we then expose them to the attenuated strain of the virus and continue treatment for about 20 days. These folks come to the hospital every day, they give blood samples every day, they see a physician every day, nobody gets too sick, and then we collect the blood samples, and it's actually very cool because we're sitting about right here. We're about day 48 post-dosing. And what that means for us is that the first 30 days, when you do this experiment, run this clinical trial, the first 30 days is where all the action happens. That's when the individuals are viremic, that's when the symptoms appear, that's when the symptoms typically clear. So we've collected all of those samples, they've been sent out to the analytical labs. We're getting the answers to the questions, right? What's the blood concentration, what's the virus concentration, and we're compiling all of that to give to my safety review committee. The safety review committee has been asked to answer 2 questions. Is it safe to give ISLA 101 to these individuals? And do you see any hints of activity? Do you see any signs that ISLA 101 has anti-dengue activity? And if the answer to both of those is yes, then we're going to move to the phase 2B cohort, which is a therapeutic cohort. The therapeutic cohort, we enroll individuals initially, we infect them first with the virus, wait till about day 7, a time at which we know they're viremic, at a time we know that they're going to be symptomatic, and then start giving ISLA 101 and see if we can treat the virus or treat the infection. We would look at that by seeing if we can enhance the rate of decline of the virus in these individuals, speed the rate of clearance of any of the symptoms. It would be fantastic to get data from both of these. We're really excited about it. Stephen Thomas, who's on our scientific advisory board, also is head of infectious diseases at SUNY Upstate. And there's a lot of questions about a challenge study, right? Is it ethical? Why on earth would people do this? Who enrolls for this? All of this stuff, understood. And I like what he says, when there's no available animal model, and for dengue, there's not a great non-human primate model. You can infect non-human primates, but they don't get sick. So you administer a mild, safe form of the disease in a healthy person, in a safe, well-controlled environment with a small number of people. You get information that normally would involve going into the field and exposing many more people with very expensive clinical trials. So we really like this. And this is just a snapshot of data from one individual. And it's a mirror image of the data that is in my control database right now that we've obtained from the Army. But if you look, this is days post-infection. Look at the blue line. This is virus level in the blood of these individuals. You can see that by day 4, the virus levels increase. By day 10, they peak. By day 14, it's gone. This is very reproducible. You can measure fever. You can measure antibody response to the viruses. You can measure signs and symptoms associated with an infection. And we're doing all of that. This is all done, right, in our study. So you could imagine, if you would imagine what a positive result for us would look like, a reduction in that virus curve, right? We would see reduced area under the curve or reduced number of symptoms or severity of symptoms. So we're excited about that. We had an announcement out recently. We'll be talking about these data in the coming weeks. We're getting pretty close. Quickly, I'm going to move to galidesivir. This is our pipeline expansion strategy. We've identified a very exciting small molecule called galidesivir. It is being developed by a company in the United States called Biotech. Galidesivir has wide antiviral activity, but the viruses we're very interested in and where they've spent the most money is in Ebola and Marburg. These are terrible, horrible hemorrhagic fevers for which there's no treatment. The US government sponsored that work with about $70 million of funding to galidesivir, to Biocryst. Biocryst hasn't spent any of their own money. As it happens, when they were running these studies, so galidesivir's had 2 phase 1 clinical trials, they've had multiple non-human primate studies that have already been conducted in Ebola and Marburg with great results that I'll tell you about later. If you come and ask me, I'll be happy to tell you about this. But they ran into COVID and the government pulled all their money. And so they stopped development. Biocryst has since become a rare disease company, and so when we contacted them, it was off strategy with no money. So we've been able to get a binding letter of intent for an option to acquire that asset, and we're working feverishly to get that wrapped up. Huge commercial opportunities. I think we can probably see where that is. I know that I'm out of time, so I'll wrap it up. We recently raised $3.5 million from some fantastic investors. Many of you will probably know who our investors are. I'm really happy to have them. And I'm happy to announce that Phil Lynch, as of yesterday, is our new executive chair. I couldn't be more excited to work with Phil. So I'll stop right there and take any questions.
So I think you said that some of the vaccines being developed are sometimes dangerous. The incoming Secretary of Health and Human Services is going to love you, isn't he?
That's not what I mean. That's not what I mean. So not vaccines in general, right? No, but the vaccines, I'll tell you the reason for that real quickly. So there's 4 strains of dengue. And what happens is the first time you get dengue, you become immune to the first strain that you were exposed to. But you develop really an inefficient immune response against strains 2, 3, and 4. And so when strains 2, 3, and 4 infect you, you mount a strong immune response, but it's ineffective. And the virus continues to propagate like crazy. And this is when you run into problems with hemorrhagic fever and death. It can be lethal. And that's actually what some of the early vaccines were doing, is they would actually protect you from one strain and predispose you for substantial problems with one of the other strains. They're really probably, Sanofi had a vaccine where they had significant problems. They just pulled it off the market.
So all 4 patients have been challenged in that phase 2 study?
Yep. All 4 patients, yeah. So it's a small number of individuals. The reason we can do small numbers is we have the Army control data behind that. All 4 have been, they've been challenged, they've been treated, and they 45.
So you compare that to some degree to what Walter Reed's already done. So
Yeah, so the 4 individuals are randomized, 3 on drug, 1 placebo. So we're going to have 1 individual, you could imagine, unfortunately for them, that we expect to have dengue. Right? We think they're going to have dengue that we could detect, and we hope that we have 3 that have no dengue or no viremia or reduced levels of it.
Dengue is it's always been a big deal. What else is in the market? What else is in clinical trials, in a competitor? And also, as my final one, how would this be given to people? Is it like a pill and injection, something like that?
Yep. The last question, this is orally available. So it's pills, right? These guys are taking it twice a day right now. So that's the answer to that. What else is in development? So we always look at vaccines, notwithstanding the caveats we just talked about. But people are working on those. Takeda's working on a vaccine that's gotten approved in Brazil and in Europe. It was submitted to the FDA in the United States. They pulled it. So there's something going on there. I don't know. And it has activity against dengue 1 and 2, not 3 and 4. So it's not ideal by any stretch of the imagination. J&J had a phase 2 small molecule. For those of you who pay attention to this, they announced that they were abandoning that program about 2 months ago. Actually, pretty nice data. It's phase 2 asset. And so they're no longer developing it as a part. I know these guys at J&J, and it's a part of the strategy at J&J. They're getting rid of all of their antivirals. They're no longer doing that. It looks like Novartis also has a small molecule. It's hard for me to find anything about that. Another company, Atea, was developing a small molecule. They failed in the clinic. That's it.
One last question, just from Pete.
How does the antiviral dose compare to the anti-cancer dose that was used, and what was the mechanism for the anti-cancer effects?
Yeah, so the anti-cancer mechanism is a little bit of a black box. There's a lot of speculation about that. The dose that we're using in this is about 1/10 the dose, the 10th, the maximal dose that people have used. People load, we're doing 600 mgs per meter squared per day. They are dosing at 4,800 mgs per meter squared per day.
Okay.
They're loading people up with this.
Yeah.
Yeah. So we're substantially lower than what's been done before, which makes me very happy.
Yeah.
Right.